Good day, everybody. Welcome to the agricultural section of the biodiversity conference. I want to introduce myself. I'm Brian Scheffler. I'm with the USDA in the United States. And Anna Childers is our co-host today, and she is also with the USDA. Today, we have a very interesting section on agricultural genomics. But as we all know, agriculture has been leading some of the most important genomics aspects, especially going into the pangenomes. And so we have some interesting speakers today, especially on complex crops of polyploids and animals. So we have Derek Bickhart, who will be talking about the work on cattle and cows. Sheena Sim. And Sheena, I apologize, I can't remember what you're talking about today. David Bertioli should be joining us on peanuts. And Avinash Shridi, and I practice this, sorry, Avinash. Sri Dasiam is from Hudson Alpha, and he'll be talking on cotton. So let's start with our first speaker, please.
So I'd like to start out my talk by thanking the conference organizers for giving us an opportunity to present. Today I will be talking about a consortium that Tim Smith and Ben Rosen have formed in order to generate pangenome resources. But in order to show you why we think that this is necessary and why we think this is the right time, we will have to actually go back in time to 1999, where DNA sequencing was still in the process of completing the human reference genome. And this is a picture of the Baylor College of Medicine with multitude of Sanger sequencing machines. In order to complete the reference genome, they needed huge production scales on the order of hundreds of machines. And they had to build the software themselves in order to generate the assemblies. At the time, each machine's daily throughput was about 2 million or 2 megabases per day, which was an astounding amount at the time, but has been superseded by recent technologies. Fast forward to today, and with a machine that fits in the palm of your hand, you can generate the equivalent of 6 months of runtime of all the instruments on that previous slide in about 48 hours. This highlights the general trend that sequencing technologies have been getting more efficient, less expensive, and have produced longer and longer DNA sequence reads. In fact, these reads are now on the order of 40 to 1,000-fold longer than the original Sanger sequencing technology. What this means in practical terms is the fact that assemblies for references can now be longer, and we actually have longer contiguous sequence, or what we term contigs. I'd like to highlight this with a real-world example, and this is the cattle reference genome assembly that was selected shortly after the human reference genome was completed. Using those large-scale sequencing farms at the time, we had started this project in earnest. And to give you a better sense of how assemblies have improved over time, I have included two graphs. The graph at the top of the page shows the assembly length in terms of billions of bases, and the graph at the bottom shows the number of contigs or contiguous sequences of that assembly. In the top graph, the closer it is to 2.8 billion bases, the better. On the bottom, the smaller the bar, the better. So in 2004, they released the original draft of the assembly for early adopters. And in subsequent years, they improved the assembly using increased amount of sequence and improvements in algorithms. However, the largest improvement has been most recent with the adoption of a third-generation sequencing technology from Pacific Biosciences and the development of the ARS-UCD 1.2 reference genome. Even further, we've taken great steps towards improving assembly algorithms and assembly sequencing experiments, a method that we partnered with the NIH-NHGRI Genomics Information Center, led by Adam Filippi. We actually took advantage of trios where parents of hybrid offspring can be used to separate sequencing reads. Our assemblies were actually able to be separated into the parental chromosome haplotigs for each assembly, and the graph on the right shows the comparison of an Angus and Brahmin assembly with a less than 1% haplotig switch error rate. A side benefit of this method is the increased continuity of the assembly. So showing the Angus and Brahmin assemblies here with a graph of the number of gaps in the assembly, we can see that they have far fewer gaps, which is the best outcome, than even the current cattle reference genome, which is ARS-UCD 1.2. Another hybrid trio binned assembly that we had generated from a yak and Highland cattle cross was even better, with far fewer gaps, and was actually one of the lowest gap assemblies that had been released at the time. So you can see that now we have gotten far better at generating more assemblies more efficiently, but what are we going to do with all these resources? And this is where our consortium plans to produce, or at least stimulate, the development of pangenome resources. But what is a pangenome? The term originally came about in microbiology, which basically takes the entirety of the genetic sequence and genes of a species and puts them all together in a resource. In our expanded definition, we're taking a look at all of the genetic variation and structural variation within individuals of the population, in this case, the bovid. And population is key here, which is why we're focusing specifically on bovids with the aim of improving cattle references and cattle resources. So Tim, Ben, and I formed the Bovine Pangenome Consortium. This is a picture of our webpage on the right. It's an international consortium that involves the buy-in from 55 institutions from 18 different countries. And we already have secured $400,000 internal funding for the sequencing of 16 to 20 trios. While we aim to sequence as many individuals using our trio binning method, we understand that many of these breeds are exceedingly rare, and we are more than happy to work with international collaborators to sequence rare and diverse breeds using the latest and third generation sequencing technologies. The ultimate goal is to create a larger resource for the community and also to sequence and assemble breeds that otherwise would not receive notice due to a lack of resources or lack of attention. Already, I've shown you two trio binned assemblies from an Angus Brahmin and Yak Highland cross, but I'm happy to report that our consortium is already working in earnest to sequence Goward and Piedmontese cross and a Bison and Simmental cross. We also have many other crosses planned in the near future, and we're hoping to work with other partners and stakeholders to sequence more as time goes on. As a bioinformatician, I tend to like to know what I'm getting myself into. So I took a look at other resources as to how big the pangenome could get for cattle, and looking at humans, where a lot of work has been done. A comparison of Han Chinese assemblies versus the human reference genome found that the Chinese typically have 30 megabases of missing sequence from the human reference genome. This increases in order of magnitude when you look at African populations, which are more diverse. So working with Bob Schnabel at the University of Missouri, we took a look at a subsection of cattle resequence cross of cattle that have been resequenced from Holstein and Angus. And just taking a brief look at a smaller subset, we identified 8 million bases that are missing from the cattle reference that you would not detect if you were aligning reads from these breeds to the cattle reference. So what does this mean on an individual basis? It actually means that 600,000 bases on average in the average Holstein or Angus is missing from the current cattle reference and would not otherwise be detected. And this highlights the need for a pangenome resource, we feel. So we feel that this is the right time to approach the community and solicit assemblies. The technology is there, we just need the organization. And we think that more diverse genomes are the solution to improving our resource and assembling more sequence that can be aligned to and used in genetic variant discovery. We're more than happy to accept new collaborators and new stakeholders, so please feel free to contact me or the other co-chairs, Ben and Tim. And we have a website that contains further information about our efforts. So I'd like to thank all of our collaborators, the other BPC co-chairs, and our steering committee. We also have many government and academic collaborators, particularly from the NIH, NHGRI, University of Adelaide, and the University of California, Santa Cruz. We'd also like to thank everyone who reached out to us for our consortium. We hope that we can work with you in the near future and improve the cattle reference genome. So thank you very much.
Thanks, Derek, for that great talk. Would you like to explain a little bit about how you see this impacting agriculture, production, and that sort of the greater impacts on cattle diversity in the future?
Yes, there are several potential impacts, and the first and probably most the probably first one will be on genomic selection. Much of the reason for this is that there are actually regions of the cattle genome present in dairy and beef breeds that aren't represented on the current Hereford reference genome. We expect that some of these unassembled regions will be included in genomic selection in the future and may result in improvement of other traits. Also, what we're hoping to do is to see specific traits that are unique to individual breeds. Several breeds do have unique characteristics that currently aren't on the reference genome. And by screening this diversity, we're hoping to pick these up and perhaps introgress them into working dairy and beef breeds in the future. So diversity is a big benefit here.
Wonderful. Well, we're hoping to have more time to ask some more questions at the end of this session. But for now, let's go ahead and proceed with Shenna's talk on chromosomal insect assemblies. Thank you again, Derek.
Thank you.
Thank you so much for giving me the opportunity to share some of this work that we're doing on chromosome scale fruit fly genome assemblies for the purpose of investigating genetic sexing. At our research center, one of our main areas of focus are tephritid fruit flies, which represent a family of insects that are highly invasive and destructive agricultural pests. Some species, or the species that I'm presenting today, is the Mexican fruit fly, Anastrea falludens, a tephritid native to Mexico and throughout Central America. It's a major pest of citrus in the citrus growing region of southern Texas in the United States, and they are largely managed using integrated pest management techniques and the method that my research program is focused on, which is the sterile insect technique. So very briefly, the sterile insect technique is a process in which male flies are made sterile, usually with irradiation, released among the wild and fertile population. And if these sterile males are lucky in love, they will result in wild females that do not produce offspring, which leads to an eradicated or suppressed wild population. This process is facilitated by a genetic sexing strain, or GSS, where males and females can be separated by a selectable phenotype to enable male-only releases. The MexFly GSS, in the MexFly GSS, males have a wild-type brown pupa phenotype in contrast to females, which have a mutant black pupal case. In this species, sex linkage is caused by an artificially induced balanced translocation between an autosome and the Y chromosome, which fixes males in a heterozygous state at the genetic sexing locus and confers a wild-type phenotype. It's because of these reciprocal translocations that we're interested in achieving chromosome scale assemblies to identify structural variants for the purpose of creating improved GSS in MexFly and new GSS in other tephritid species. This and many other genomes were sequenced as part of the Ag 100 Pest Initiative, an undertaking by the USDA where we are sequencing the genomes of 100 pestiferous and beneficial insects. You can track our progress here, and if you want to learn more about this project, Anna Childers will be presenting on this also at this meeting. So to sequence the MexFly genome, we began first by extracting high molecular weight DNA from one male wild-type MexFly To do the extraction, the thorax was separated from the head and abdomen, and only DNA from the thorax was used to prepare a hi-fi library, which was sequenced on a PacBio Sequel II using 2 SMRT cells. The reads of which, the subsequent reads from this sequencing were assembled using the program hi-fi ASM. And by using only the thoracic tissue, we were able to use the other body parts to make a Hi-C library, which I'll discuss later. And also using the thoracic tissue, we were able to eliminate a lot of the off-target microbial sequences from the gut microbiome. So this is a visualization of the genome where the circumference represents the genome size, which is about 830 megabases, and the lengths of the scaffolds, or in this case contigs, because this is a contig assembly, are aligned in descending order. So this represents like the length of the longest scaffold, which in this case is about 100 megabases, and also really close to basically the size of a chromosome. The initial contig was made up, contig assembly was made up of about 200 pieces, and 16 of those largest pieces represented 90% of the whole genome. Lastly, an analysis of gene completeness based on a benchmark of universal single copy orthologs, or BUSCOs, revealed that the genome contained almost 98% of the genes that we would have expected in a Diptera species. Following the contig assembly, we applied chromosomal contact conformation mapping derived from Hi-C sequences to scaffold the contig assembly into a chromosome scale. To do this, we applied we used the 3D DNA pipeline to identify the contacts between contigs and visualize these contacts so that way we can make manual edits using the program JuiceBox. This is a heat map showing the density of contacts between reads, where higher saturation represents more contacts. Pixels within the green boxes are contacts between the sequences from the same contig, and within the blue boxes are contacts between sequences from the same scaffold, or in this case, the same chromosome. As you can see by the size of some of these green boxes, in some of the cases, entire chromosomes were assembled at almost entirely from only 1 or 2 contigs. After assembling the contigs into scaffolds, a much larger proportion of the genome ended up in very few large pieces. So this was assembled into 5 autosomes in the X chromosome. After scaffolding, there were still about 150 contigs that remained unplaced, but they only represented about 5% of the genome, so they were very short contigs to start. We applied dd-rad sequences derived from several mapping populations of 2 different genetic sexing strains of MexFly to do a QTL analysis so that we could identify the loci that are most tightly linked to the black pupae gene. And the QTL analysis showed that the black pupa gene is located on one of these autosomes. I wonder if you could tell which. It's where these peaks are. But the interesting result here is that in the 2 different strains, they map to slightly 2 different places within the same chromosome. This is likely due to the origin of sexual dimorphism in this trait, which was caused by chemical mutagenesis. And chemical mutagenesis is a technique that induces the creation of structural variants such as translocations and inversions. So in addition to creating a balanced translocation that I described before between the autosome and the Y chromosome that causes the sex linkage, the black pupae gene may also be located in a strain-specific or otherwise polymorphic inversion. Now that we have a chromosome scale assembly, future work will be pursued to investigate which of these conformations is the wild type. To validate our assemblies, we relied on the conservation of molar elements in Diptera by performing an analysis for synteny with Drosophila. And this was performed using the same BUSCOs that we use to assess the completeness of the genome. So in this visualization, the lines represent nearly 2,800 genes in the Diptera BUSCO dataset. And as you can see, you can easily identify which of the MexFly chromosomes on the top are homologous to which Drosophila chromosome arms on the bottom. And though we see very high conservation of gene composition of these molar elements, Collinearity between Drosophilids and Tephridids appears to have decayed over the 150 million years since they last shared a common ancestor, as indicated by the genes that appear to have undergone both intra and interchromosomal translocations between the evolution of Drosophila and Anastropha. Comparing the assemblies of two Anastraphas, where now Mexfly is at the bottom and the Caribbean fruit fly Anastropha obliqua is at the top, you can see a very high degree of collinearity, which demonstrates both the accuracy and conservation of gene order between these two very closely related species. Here I plotted the start positions of shared buscos between Mexfly on the y-axis and Caribfly on the x-axis for the contig that represented almost an entire chromosomes for their respective species. They're highly collinear, save for this one region that is indicative of a chromosomal inversion. And if you look at the GC content of their 5' end, there are signatures of it representing heterochromatic sequences with an elevated GC content in both of these contigs. This could be indicative of a telomeric region of the chromosome, but further validation will be required to confirm this. So, in summary, because of our interest in genetic sexing strains and the chromosomal translocations that they harbor, we've been developing methods to achieve chromosome scale assemblies. We did this by using the thorax of one fly to generate a contig assembly, and then using the rest of the body segments of the same fly to create a high C assembly for scaffolding. The subsequent genome assembly or chromosome scale assembly was then used to perform a QTL analysis to identify the autosome and gene region which harbors the causative gene for the genetic sexing trait black pupae. And we validated our assembly using synteny, where we saw a large amount of collinearity between closely related species, which indicates the accuracy of both assemblies, and hints of telomeric regions, which indicate that we are approaching a telomere to telomere assembly for these chromosomes. With that, I would like to thank our funding sources, USDA, ARS, APHIS, and NIFA, all the people listed on the slide who were the ones who did all the real work. And lastly, I would like to thank you for your attention. Thank you.
Thanks, Shenna. That was a really great talk. And thanks for also the plug for the Ag 100 Pest Talk that will be tomorrow. One of the questions that came in, did you compare your genome to the olive fruit fly genome? And can you comment on that? I know you've been working hard on a lot of synteny work, and you've got other genomes planned, so perhaps you can include that in your discussion.
You know, that's a really great point. I haven't actually looked at the olive genome, olive fly genome. I know that it is at a chromosome scale. It was done with linkage mapping. I would expect that there would also be a high degree of synteny. I've looked at synteny between our species, Anastropha and Zeugodacus, which used to be Bactricera. And there's a high degree of synteny there. You see a little bit of gene shuffling over the course of those, the evolution of those two lineages. But yeah, I would expect them to be highly synthetic. However, I would also expect a lot of maybe gaps, depending on, so I know that was an Illumina assembly, which means that it's not going to include a lot of the gene poor regions or maybe the AT-rich regions of the genome. So in comparison with other Illumina chromosome scale assemblies that I've looked at, that's what I would expect. And so, yeah, I look forward to looking at that definitely.
Wonderful. Well, like we said, we'll hopefully have some more time for additional Q&A at the end, but I'd like to go ahead and move on with Avinash's talk now. Thank you.
Hello, everyone. My name is Avinash Sridasyam. I'm a computational biologist in Jeremy Schmutz's group at Hudson Alpha. And today I will talk about our work on understanding the genetic variations between 3 elite cotton lines. So firstly, a brief introduction about cotton. It is needless to say that cotton is one of the most important crops, based on the amount of textile we use that comes from the cotton is about 60% all over the world. I think it suffices to inform the importance of this cotton crop. And moreover, cotton is one of the earliest domesticated non-food crops, and it has been independently domesticated at least 4 different times in both the Old and the New Worlds. And the very first domestication event occurred about 7,500 years ago in Indus Valley of India and Pakistan. And the species was Gossypium arboreum, which is a diploid wild tree. And the next event occurred in Arabia and Syria, and it's a Gossypium herbaceum. And the next 2 events occurred in Mesoamerica about 5,000 years ago. The 2 are Gossypium hirsutum, the upland cotton, and the Gossypium barbadensis, the Pima cotton. These 2 are allotetraploids. So it is important to note that most of the cotton produced in the world today is the New World species, that is Gossypium hirsutum. So besides those species which I just mentioned, cotton has over 50 different species, and 7 of them are allotetraploids and 45 of them are diploids, and they're found all over the world in Africa, Asia, Australia, and America. And out of these species, the 5 of them highlighted here are group sequenced and published in Nature Genetics in the early 2020. So here are some genome stats for these species. The genome assemblies were up to 2.2 to 2.3 gigabytes in size, and the contingent 50 range from 780 kb to all the way up to 10.1 MB. And the annotated protein coding genes range from 74,000 to 78,000. So we tried to understand the variation between these allotetraploids and accounted for the SNP variations, which was about 4 to 13 million in a 2.3 kg genome. That is about 0.19 to 0.53% genome differences between these allotetraploids. And the SNP rate accounts to about 2.2 to 6.5, which is not much really. And the indels also represented about 3.3 to 5.5 MB. So as I've said, this is not much. To give you a context, we also analyzed a subspecies of Panicum hallei, 2 subspecies of Panicum hallei, where we saw the SNP rate of about 7.1. And between the natural accessions of Brachypodium distachyon, a model grass plant, we saw a SNP rate of 4.6. So this clearly shows that there is a very low genome variation and high stability. So the next question pops up is, how would we go about improving this cotton crop with so little genome variations? So to address this, the very first question we asked, or in fact Don Jones asked, there are other elite varieties grown by breeders both in US and elsewhere in the world, which are high fiber quality and high yielding varieties. So what is it that makes them elite? So that's the main question. So to address this, Don picked 3 different cultivars, elite cultivars. One of it is UGA 230 from Peng Chi at the University of Georgia, which is called Southeast Line, and UA 48 from Fred Borland's group in University of Arkansas, and CSX 8303 from Warwick Stiller at CSIRO, and this is called Australian Elite Line. So how are these elite? So from this table, this shows the metrics like lint yield. MIC is the micronear, which indicates the fiber firmness and maturity, and UHM and seed bowl, and also the aggregated quality, weighted aggregated quality score clearly indicates that these are elite lines based on the mean calculated across 32 different elite lines grown in 11 different locations. And besides the high quality and high yielding, CSX 8303 has a distinct ochre leaf line, which is incompletely dominant over the normal leaf. Here we can clearly see the ochre leaf shape for CSX compared to the TM1 reference line. And the UA 48 is the early maturity cultivar with a short season. and UGA 230 on the other hand is an indeterminate or full season cultivar. So what is it in their genomes that makes them alive? To address this, we have sequenced these elite lines, and the contigent 50 is 8.1 to 29.9, which is very high quality genomes. And besides that, we have also regenerated the assembly for TM1, that's version 3.1, and the contigent 50 is about 40. So, and annotation stats look good as well. I want to note that these 4 genomes are still not published as of now. And then we assess the gene content among these elite lines and also compare that with the other allotetraploid cottons and see the orthologous genes or the syntenic blocks of the orthologous genes is remarkably highly conserved across these chromosomes and the subgenomes. And we then try to identify the unique and shared gene families between these elite lines, and we did not find any elite line specific gene families with biological relevance or functional role related to the cotton fiber traits. And then we looked into the expansions in these gene families. We saw expansion of 9 to 19 families within these elite lines, and UGA 230 with 18 gene families and 324 genes showed enrichment for oxidation reduction processes, and whereas UA48 was enriched for tubulin complex assembly and auxin transport. Tubulin complex assembly is again a fiber-related trait related genes. And in case of CSX8303, we see enrichment for histone, H3 kinase methylation, as well as trichome differentiation, again a fiber-related trait genes. And these are the dot plots showing the nucleotide alignment, which clearly shows there is a high genome collinearity between these elite lines with very few variations. Highlighted here are variations specific to CSX8303. In case of A06 chromosome, we see a tiny inversion, and in case of A12, we see a duplication event going on here. So these are the signatures of CSX8303. And to understand the differences between elites and the TM1, we assess the SNPs and indels. And this plot, Circus plot here, shows the densities and the signatures of specific elite lines. Highlighted here are, in green is CSX8303 versus UA48 specific signatures, and the red one is CSX specific signatures. I want to bring your attention to this highlighted portion in A08. We clearly see a very high SNP and indel rich regions in the A08. And again, we did not find much of a SNP variation. The SNP rate is about 0.4 to 0.6, accounting to about 1 to 1.7 million SNPs overall. And these circus plots are similar to what we just saw in the previous slide. The signatures are pretty similar, but I want to again point out that the A08 SNP and indel region, we did not see that in these elite lines compared to the between the elite lines. So this clearly indicates that there are stark differences between elite lines and the TM1, and using TM1 as a reference for genotyping may not be much efficient and likely not as good for breeding efforts either. So it's important to generate reference sequences for these elite lines as well. And as expected, the SNP rate between these elite lines is very low, 0.3 to 0.59 SNP rate. So besides the SNPs and indels, we also looked into the large structural and sequence variations between the elite lines, and as the structural variations being copy gains, copy losses, and translocations and inversions. So these rearrangements are important because these rearranged regions are inherited differently compared to the syntenic regions. So, but we did not find any co-localized structural variations except for the inversions highlighted up here. So to understand the differences in the fiber development between these elite lines, we assessed the transcription response at 7, 14, and 21 days post antithesis, and we saw the variation or differential expression between these time points in each of the elite lines. And besides finding the differentially expressed genes, we also clustered the genes to identify specific trends, like the upward trend highlighted in blue was enriched for supramolecular fiber organization, and the downward trend highlighted in red shows nitrogen compound transport and protein localization enrichments. So besides that, we also looked into the differentially expressed genes between these elite lines by mapping them RNA-seq reads onto the TM1 reference. And here we identified the regions which are dense and specific signatures of each of these elite lines with a hope to identify the regulatory regions that are selected for in these elite lines. And here in this graph, we see highly differentially expressed genes in elites versus TM1. And these genes are also enriched for biologically relevant biological processes here. So this work is still in progress, and we are analyzing further to identify the candidate genes which contribute to the phenotype, observed phenotype, as well as the fiber traits. So to summarize, we have generated 3 high-quality reference cotton genomes along with the new TM1 reference, and we catalog the differences between elites and the TM1, which warrants for generating new references for these elite cottons. And also we identified and cataloged structural and sequence variations between elite lines. And the transcriptional response in fiber developmental stages is being currently investigated. And it also appears that the effect of sequence variants on gene regulation could be really key. So we are analyzing those as well. And with the low genome variation and genotypic diversity, using the variation among these elite lines can be helpful in improving these lines. So these are our funding sources. And I would like to take this opportunity to thank our members at the Hudson Alpha and also the folks at JGI, and my sincere thanks to amazing cotton team. And thank you all for your attention.
Thanks, Avinash. This is clearly an extensive amount of work. What else needs to be done to understand breeding selection and also the exclusion of the various gossipiniums?
We need to understand what is going on within these breeding lines. So we are to do the resequencing of some of the selected breeding lines to understand what are really being selected for the genetic elements per se. That's in process. That's ongoing work. Thank you.
Yeah, I think it's very surprising that there's so little genetic variation between cotton. Do you have any thoughts to why that might be? Is it do you think that there's a recent divergence?
Oh, sorry. Yeah, go ahead, please.
Yeah, that's the question. I'm sorry. Go ahead.
Yeah, these are recently diverged. So within these allotetraploids, there are about 8,000 years being really going on between the selection or domestication. So we don't expect much of it, but unusually it is strange that these cottons show so much little variation. So to understand the genetics of the cottons and breeding, so we need, that's the main reason we are sequencing these elite lines to better understand what is really being selected for. So the overall, if you take about 5 million years was the time when these diploids were diverged, and these allotetraploids came together or hybridized about 1 to 1.6 million years ago. But main selection or breeding has been happening about since 8,000 years ago. So that could explain why we see so little variation.
Wonderful. Well, I hope we'll have some time for some more questions here at the end. Let's go ahead and hear from David on peanut first.
Thanks very much for the opportunity to speak this morning in this rather awkward recorded Zoom format. I'd like to tell you about peanuts and about our use of wild peanuts in improving crop. Now, wild species, they grow only in South America. They grow in all sorts of environments, but most typically they grow in the Brazilian savannas, the cerrados. They look like this. And if we look down to the bottom of the savanna floor, we can see these little wild peanut plants. We're interested in wild peanuts because they have fantastic pest and disease resistances. The focus of our talk is a species called Arachistenosperma. And you can see the contrast when a cultivated peanut leaf and an Arachistenosperma leaf are inoculated with rust fungus spores. There's a similar story for late leaf spot, which is the most important peanut disease in the world, for fall armyworm, and for root knot nematodes, which is the focus of this talk. You can see that the cultivated peanut root has lots of these red stained egg masses. The wild Arachistenosperma has absolutely no nematode replication at all. Now, the accession that we use is an accession collected in 1987 by José Valls. It's called V10309. He collected it near Rondonópolis in Brazil, and he returned to that location in 2004, and this is what he found. He could still find some plants growing under this mango tree at the side of the road. Returning again in 2019, this is what's found. This is the same concrete pad here. The mango tree has been cut down, and there are absolutely no plants to be found. So this accession, this particular genetically unique accession, has become extinct in the wild. Thank goodness that he collected it. Now, the wild peanuts, they are almost all diploid, and cultivated peanut is tetraploid. The origin of peanut was a diploid species called Arachis hybridized with a different diploid species, Arachis. They contributed the A and the B genomes. There was a polyploidy event to produce the polyploid, the tetraploid peanut. And you can see the chromosomes highlighted with fish nicely here. In order to provide a genetic framework, initially we sequenced the diploid ancestors to avoid tetraploid genetics, the complexities of it. That was published in 2016. Subsequently, with PacBio sequencing, we were able to sequence the cultivated genome itself. That was published last year, also in Nature Genetics. All of these genome sequences, they provide excellent references to work in, and they make things tremendously easier. But going back to using the actual species, because Arachistenosperma is diploid and cultivated peanut is tetraploid, before we can incorporate it into crossing schemes, we need to make Arachistenosperma into a tetraploid form. What we did in this case is we crossed Arachis bachizochoi, which is a sort of B genome, with Arachistenosperma, the A genome. This creates a F1 sterile hybrid. We treated with colchicine. We literally just take cuttings off here, each one about 20 centimeters long, soak them in colchicine, rinse them, plant them. After some time they recover, and after some time more, some of them produce pegs and seeds. These give us the neotetraploid, which is crossable with peanut. We cross this neotetraploid with elite peanuts, produced a population of 230 individuals, which we inoculated with the root knot nematodes. We did a bioassay. We genotyped, so we had complete genotyping and phenotyping, created a map, and identified QTLs. We identified 2 QTLs, both very strong, one at the end of chromosome A2 and one at the end of chromosome A9. Those are both very strong. This is the effect in suppressing nematode replication, and these scales here are log 10 scales. Well, because we've got it mapped on a genome framework, that automatically gives us markers, and we went straight into a back-crossing scheme with marker-assisted selection. Through 3 generations, arriving at backcross 3 in 2019, that gave us about 250 plants. They were again genotyped with a SNP array, a 48,000 SNP array. So we have the genetic profile for every single one of those plants. This is a snapshot from that, and we can visualize the wild chromosome segments as blue segments here. We were able to identify which ones had the nematode resistance QTLs, and we were able to confirm the resistance. These plants, they also grow fantastically. They've been crossed into an elite background, and they grow in an elite way. Here's a field of them that is this year. These are elite genotypes that are grown in Georgia. These are the backcross 3 plants. You can see they're vigorous and large. They also produce pods, which are the same format as the elites, and with the same seed size. They really are very, very nice looking plants. So overall, the wild peanut species, they harbour fantastically strong pest and disease resistances. Understanding the species relationships between the wilds and the cultivated, which is A genome, which is B genome, etc., and how they are crossable. That streamlined our crossing schemes, and having a genomic framework that has speeded tremendously our ability to map and introgress. And I've given an example here of how we did that with, for root knot nematode resistance. Of course, it involves teamwork. Thank you to all those people involved. I hope I haven't missed anyone. And of course, nothing would happen without our funders. Last of all, thank you very much for your attention.
Thanks, David, for that great talk. Could you discuss a little bit how hard it was to sequence a polyploid genome?
Yeah, sure. Well, initially, that's why we sequenced the diploids, because we did a simulation with Illumina Go sequences, and they collapse. So we built up the diploids first. Then with PacBio, it really is a different thing altogether. Sequence B genes, they can easily be sent similar, but PacBio solves it, and it dropped out beautifully. Now these PacBio genomes are amazing.
Well, the advancements in technology have really clearly enabled all of these projects, and really some wonderful and beautiful projects here. Unfortunately, we don't have time now at the end of the session for more questions. There are some great questions in the stream, and hopefully we can take those questions and our speakers can join in to the poster session where we can have some more discussion on these topics. I want to thank each of our speakers again, as well as thanking all of our audience for their participation.